Our presentation is focused on the effects of alcohol abuse and its effect on gene expression in the brain. The study looks to identify whether or not widespread gene expression may be the cause caused by alcohol abuse, which may lead to drug dependence. The study found that alcoholic brains had high expression of endogenous retrovirus and genes with high GC content association with DNA hypomethylation and increased histone trimethylation, suggesting that epigenetic mechanisms play a critical role in alcohol addiction. Histone trimethylation is a process where 3 methyl groups are transferred to amino acids of histone proteins. DNA hypomethylation is the addition of methyl groups to the C and A DNA nucleotides. To determine mechanisms of underlying brain pathology, transcriptome profiling was used. In recent microarray studies, it was found that individual genes could be a cause for addiction phenotypes. There are 3 key findings in brain transcriptome. The first is that individual populations of neurons and glia are categorized by unique transcriptional signatures that show the current functional state of the cells. The second transcriptome for complex tissues, such as the brain, are organised into networks of co-expressed genes. And finally, these networks of co-expressed genes reflect functional and structural organisations of the tissue in the brain. These help to understand the brain transcriptomes and allow interpretation of differential expressions of the genes associated with the pathology of the brain.
Using post-mortem brains of human alcoholics, gene expression levels were profiled and compared to control cases. This was done using a systems approach to DNA analysis, data analysis that combined differential gene expression, gene co-expression networks, and cell type. Critical epigenetic components of gene co-expression were identified as a central role for epigenetic regulation in gene regulation in alcohol-induced changes in global expression. This allowed the researchers to generate a hypothesis of changes in the brain of alcoholics that linked epigenetic regulation of gene expression with cellular abnormalities that were previously identified. The results could lead to treatment strategies in alcoholic brains and could be a prototype for analysis of existing microarray data. Autopsy brain samples were used. 17 alcoholics and 15 matched controls were used from 30 males and 2 females. Cases were matched by age, gender, post-mortem interval, and brain pH. Agonal hypoxia did not differ significantly between cases, and none showed hypoxic encephalopathy. The techniques used in this research include microarray profiling and QRT-PCR validation, weighted gene co-expression network analysis, and chromatic assays. The chromatin assay was used to quantify global H3K4 histone methylation. The results found that alcohol abuse is associated with widespread changes in gene co-expression. History of alcohol abuse was associated with global changes in gene expression in all 3 of the brain regions analysed. The results confirmed previous results in other studies showing changes in brain expression in alcoholics. Upregulation of GC-rich genes in alcoholics was found to be associated with increased H3K4 trimethylation in all 3 brain regions. This may be due to increased levels of H3K4 trimethylation in promoters. Alcoholic-induced upregulation of genes was also found to be involved in transcription co-repressor complexes. Methyl CpG binding proteins MBD3 and chromodomain helicase were found to be highly upregulated in alcoholic brains. These proteins are partners in the nucleosome remodeling and histone deacetylation transcription of co-repressor complexes that is involved in transcriptional repression. Most other members of the transcriptome co-repressor complex were also upregulated in alcoholics. This suggests that these complexes are activated and play a role in downregulation of some genes in alcoholic brains. It was also found that alcohol abuse can change gene expression through chromatin changes. This causes researchers to focus on genes involved in epigenetic regulation of gene expression. Alcoholic brains and cocaine addicts have also displayed an upregulation of the prioritized gene MBD3 and others. This is a TCC protein which is very important in the coupling of HDAC activity and in chromatin remodeling. The upregulation in synaptic genes was detected in the cortex, which may indicate an increase in glutamatergic transmission in response to the brain cell degeneration. This could explain the long-term impairment of cognitive control in alcoholics.
This study is the first to demonstrate widespread changes in DNA methylation in the brain of those who are alcoholics. where it may then cause the development and maintenance of alcohol dependence. Chronic alcohol abuse can result in a decrease in DNA methylation, resulting in a decrease in S-adenosyl methionine, known as SAM. As SAM is increased, its metabolites decrease, S-adenosyl homocysteine and homocysteine, in long-term alcoholics, which may be the cause of DNA hypermethylation. Also, an increase in gene-specific trimethylation of H3K4 was found. Activation of multiple genes involved in transcription co-repressor complexes was also found in the brain of alcoholics. DNA hypermethylation can also cause transcription activation of HERVs in alcoholic brains. The consequence of HERV activation is an ERV-encoded glycoprotein, synctin, which can activate microglia and astrocytes. This leads to brain inflammation in alcoholic brains. Microglial activation can also cause the degeneration in brain cells. The synctin-activated astrocytes secrete compounds which can cause cytotoxicity in oligodendrocytes, as well as degeneration of the myelin sheath. There was an unexpected finding within this study. DNA-binding proteins, which are part of chromatin modification complexes, preferred to bind to GC-rich areas compared to low GC areas. The upregulation and downregulation of these genes in the brains of alcoholics could be determined by these DNA-binding regulators. To conclude, the evidence provided from this study suggests that widespread changes in gene expression is mainly caused by chronic alcohol abuse. These changes are caused by changes in the chromatin state. As a result of this study, epigenetic interventions may be able to correct the changes in the gene expression and functional abnormalities in the brain of chronic alcohol abusers. Only 2 alcoholic brains were used compared with 30 of the male brains. The hypermethylation could present itself differently in females, therefore an equal number of males and females should be used to avoid gender bias. Also, in general, males can consume and tolerate more alcohol than females, which should be considered when choosing test subjects. The researchers did not consider ethnicity when choosing subjects or matching controls with alcoholic brains. Different ethnicities can tolerate different levels of alcohol, so one person may consume a higher amount of alcohol than another, but may still be alcohol abusers.